Hello. Oh, welcome everybody to the BML seminar series. This is our last seminar for about a month. And I think we had a little bit of a hiatus, but coming back again in August. So we're really excited today to welcome Dr. Lilian Aoki. She's a coastal scientist studying ecosystem dynamics and resilience in nearshore and estuarine habitats. Lilian uses a variety of field, lab, and computational methods to build an integrated and cross-scale understanding of coastal ecology in a rapidly changing world. Her current projects focus on understanding recovery trajectories in seagrass meadows following marine heat waves, and the role of land management in facilitating carbon sequestration in tidal marshes. And so lots of things that I think are of interest to many people in our audience. She earned her PhD in environmental sciences from UVA. And in 2026, she was selected as an early career fellow by the Ecological Society of America. Woo! Awesome. She is currently a professor at the University of Oregon, and we're really lucky to have her here today. Fun fact about Lilian is she plays field hockey. I know, so cool. Okay, I'm gonna give this to you now. You're gonna try that.
I think people can hear me, yes? Okay, great, awesome. Amazing, thank you. Ooh, that seems loud. Okay, I'll be quiet. Thank you, Anya, for that introduction, and thank you, everybody. And I'm very, very excited. This is the first time I've been to BML, and it's been fabulous, and I had so much fun talking to everyone today. So I'm really excited to share some work with you and to chat about coastal vegetated habitats. Okay, so we're gonna get going. And I don't think for this audience I need to really introduce coastal vegetated habitats, but just for sort of the sake of getting started and framing, I'm thinking a lot about these habitats across kind of the intertidal zone. So from sort of more closer to the water with seagrass meadows, and then moving up the, let's see if I can get this, up the tidal gradient up into salt marshes. There are mangroves in this picture. I have not had the opportunity to work in a mangrove forest yet, but that will come one day. But I think a lot about the this foundation species structuring these communities. So this really strong impact of the foundation species along this tidal gradient and the kind of functions and services that these ecosystems provide. So today I'm going to go through a couple of stories. One about seagrass up in Puget Sound, so eelgrass up in San Juan, and we'll talk about salt marshes in Coos Bay as well. So that's where we're going. And I do like to start with some beautiful pictures to remind us when we say foundation species and functions and services, what do we really mean? Some things that we mean are habitat. So these vegetated systems create habitat, they physically structure the habitat for lots of different organisms, and that's sort of charismatic macrofauna of different types, but also smaller little guys, the microbes also. I didn't picture them, but they're there as well. So that's one really important service and function that these foundation species are really doing for us in the coastal zone. Another one that I think about a lot is carbon sequestration, and this has come to the forefront more and more in the last decade or so because of the potential role that these habitats can play in terms of mitigating climate change and helping us to draw down emissions and offset some of our carbon emissions. And so for that, we're thinking a lot about sediments. So we're thinking a lot about the carbon that's held in the sediments as well. And I like to sort of introduce myself, my approach as a scientist. So I really think of myself as an ecosystem ecologist. I think about structure and function and dynamics. So like sort of asking questions about the sort of biophysical properties of these habitats, the ecological processes, how they're interacting across time and space, how do ecosystems persist, are they vulnerable, are they resilient, and what kind of drives those sort of ecosystem scale properties. Okay, and I'm actually not going to talk about seagrass wasting disease, so hopefully that's okay. I know there's a lot of seagrass wasting disease people in the room, but yeah, today is some other stuff. But okay, so the other thing is the ecosystem science approach, ecosystem ecology approach. Often we love to draw boxes and arrows, and we love to draw a box around our ecosystem and say what's happening inside that and what's happening outside that, what's our flux across this boundary. I love to do this, and sometimes it's the right approach. Sometimes you need a little bit more understanding of what's going on, what those biological interactions are inside. But sometimes we can just say, here's our seagrass meadow. the black box and what's going on across that boundary. So just some sort of framing to keep in mind for these pieces. So, okay, so that's my setup. I'm going to try not to talk too fast. So I'm going to talk about these two different ecosystems. Again, we're going to talk about different functions, habitat provisioning and carbon sequestration in the two different the two different ecosystems. And I think these are two parts of my lab right now. So I'm sort of early in establishing my lab. I have a marsh side and a seagrass side, and I'm trying to bring them together a little bit and think about what bridges between them. And some of what I think is that bridge is thinking about these ecosystems. They're experiencing these stressors at this land-water interface, and this sort of integrated response to those stressors is some of what I'm thinking about. So, okay, so we're going to start with seagrass meadows. This is work that I started during my postdoc up in the San Juan Islands, and we've continued it. So this is north of us, north up in Puget Sound, beautiful place. And these meadows are really intertidal, so they come out of the water completely at low tides. So they're very exposed, and they're very exposed at midday low tide, so they get a lot of desiccation, they get a lot of stress. They're potentially very vulnerable to lots of kinds of disturbance. One kind of disturbance is heat waves. So actually, I don't know that this event was that memorable. I don't know if it was memorable in California. It was extremely memorable in the Pacific Northwest. So this was June 2021. We had what became known as the Pacific Northwest Heat Dome. This was an extreme atmospheric event. We were crossing all kinds of temperature records on land. And this is kind of an image from NASA of the extent of the event. And it's sort of centered beautifully in our study area, San Juan Islands. And what the eelgrass meadows experienced during this event was pretty extreme. So this is a plot that shows temperatures during this heating event. So during the month of June, the solid lines here, these are all sea surface temperatures, so these are all measured from remote sensing satellites. The solid line is what was observed during that time, and the dashed lines are the long-term mean for the green line and long-term 90th percentile for the gold line. So once we cross that 90th percentile, we start to get into what we consider marine heat wave territory. If it's extended over 5 days or more, that's sort of official marine heat wave definition, but these are very extreme temperatures. So these are very extreme for that location. From the satellite sensing, we're definitely in that extreme temperature anomaly. And then in the back of this plot, I know it's really pale, but the pale lines here are the hourly temperatures measured with in situ loggers, which are 15 degrees more than that sea surface temperature. So these meadows we're experiencing is a very extreme event, a very anomalously warm time for them in the meadow. And unfortunately, what this caused was a large-scale decline. So these meadows in the San Juan Islands, many of these intertidal meadows just seriously declined. So this is imagery. This is aerial imagery taken by drone with our collaborator, Bo Yang, who's at UCSC now. But we have a beautiful record. We actually started mapping these meadows in 2019. In each image, in here, the land is on this side, the ocean is on the right side of each image, so we have the same area in 2019. 2020, we have lots of coverage. In 2021, we mapped about a week after the heat wave, and the intertidal was just gone. It was just bare sand, and it really hasn't come back at this site. I don't have the imagery pulled up yet, but it hasn't come back. And we see the same thing at other sites, so pretty much everywhere we went, this is what we saw. So the orientation of the image is flipped. The land or the ocean's on the left side and the land's on the right side. So this is the intertidal zone. And again, we have this sort of continuous coverage in 2019 and 2020, and then in 2021, retraction of that upper zone. All the intertidal grass just died. So that's not great. And then, as I said, this is sort of work that I've sort of picked up again with my lab now, where we've been looking at, we're continuing these ecological monitoring surveys. We're looking at structure, so thinking about kind of classic ecology, quadrats and transects, and looking at density and also lots of other things. We do measure disease, but I'm not going to talk about it right now. But sort of looking at the structure of the meadows. And then since 2021, we've been able to also look at function for fish habitat through a collaborative eDNA project, which I'll talk to a little bit more in a moment. But we're able to sort of assess the fish communities at these sites and see sort of changes over time. And from that, hopefully infer something about habitat provisioning. So jumping into some data from this. These are all of our sites. These are kind of a lot going on in these plots, but this is basically cover and density. Each panel is a different site, and we have our time series on the x-axis, and the bars, the stacked bars are percent cover, so green is the eelgrass cover, and then we have macroalgae and bare sediment, and we can see, like, most of our sites, we have a pretty substantial decline over the time series, and at these 3 sites, basically no recovery of cover. The shoot densities are the line that's plotted, at the y-axis is on the right, and those are also pretty much little to no recovery in the intertidal. A couple of our sites, we do start to see some recovery. So a couple of our sites we started to see, we went back last year and we started to see a little bit of recovery. We're gonna be back out there in 2 weeks, so we'll be able to extend this and see if we continue to see that. But basically, within 4 years after this extreme heat dome, we don't see any consistent pattern of recovery. So these areas have not really been able to come back. And I don't think it's that surprising when you go out there and you see this just incredible dense macroalgae coverage of the intertidal. I'm not shocked that the eelgrass can't grow back. So this is images of the same area in 2019 before the heat dome, and then last year it's just this heavy macroalgae cover. And so we can think about, if we're thinking about this from a sort of ecosystem dynamics perspective, the stressor that caused the decline, this intense acute heat wave, that has dissipated, but a different stressor may have emerged, this macroalgae that's preventing the sort of shift back into the previous seagrass state. And so from a sort of ecosystem dynamics perspective, we can think about these different stressors that they may prevent an ecosystem recovery being different from what causes initial decline. And this has lots of implications for restoration, which has been a real interest in the islands. As we've seen these eelgrass declines, there's been a lot of interest in trying to think about restoration. But of course, if we have this intense stressor of the macroalgae, we need to think about how to sort of remediate that or how to overcome that if we're going to think about restoration. Okay, so that's sort of the structural piece. And then the second piece of this is to think about the ecosystem function. So we've had this sort of strong impact in ecosystem function. Can we think about connecting that to habitat provisioning? In this case, thinking about fish communities. So this is part of the PICO eDNA observatory, which I think folks here are involved in as well. So this is, we go out and we take some water samples and we look at all the genetic material that's in them and use that to sort of understand something about the fish that might be present. And I want to preface this with saying, like, I am not the person, I'm not the eDNA lead, let's put it that way. I'm the field ecologist supporting this work. But it's been incredible to be part of this, network. And I do love this figure from Jen Sunday that shows the progression from our water sample full of bits of genomic material and where we go from there. And this is also just to recognize that there's a lot of work that goes into running a big collaboration like this. So the PICO network is all up and down the West Coast. We have 17 sites, I think, 52 partners, lots of people involved, lots of people doing the extractions and the bioinformatics, and then they give me a table of taxa, and I'm very happy with that. So if we start to dig into the PICO data a little bit, this is just looking at the number of taxa that are present, and then just sort of overall number of taxa present at our different sites. Each color is a different site. We started this the year of the heat dome, so we don't have a pre-heat dome record, but we don't really see like a dramatic pattern. We don't see a dramatic difference. We see a lot of variability between sites and between years. If we dig into this a little bit more, we see a lot of species that are present at some site at some point. And we see, in addition, we see this kind of like regular eelgrass community. So there's sort of eelgrass fish that appear at least one site every year consistently throughout the data set. And then there's other fish that sort of appear a little bit less consistently, maybe at only a handful of sites, 1 or 2 sites, or maybe only 1 or 2 years. And if you are sort of a fish person, I'm also not a fish person, so this has been fun for me to learn stuff. But if you're sort of interested, like, the fish that we find consistently at all these sites every year tend to be what you expect to find in eelgrass. Pricklebacks and gunnels and stringer perch and things like that. So it's sort of like a nice gut check that we're getting what we expect to get. This is exciting because I'm just saying all the things I'm not. I'm not a community ecologist, but here's some PCOAs. So this is kind of exciting. I don't know. The data from this project has really only started to emerge recently, so we're sort of like dipping our toes into it and figuring things out. But it's kind of interesting that we see, it's the same PCOA, but just sort of plotted on the left is the temporal trajectory of each site, and then on the right, the polygons are the years, so you can kind of see the spatial and temporal patterns a little bit. But it's sort of interesting that we kind of have like two sites that are further, these two sites are closer to each other geographically. They're also warmer. They're also closer to the Fraser River, which is a big freshwater input to the San Juan Islands, and it's also a source of pollutants. It's a source of lower salinity water, so there's more salinity variation. So there is this sort of spatial grouping, maybe a little bit between our sites. And then these three sites are sort of further south, and they are a little bit cooler and a little bit less influenced by the Fraser River. And if we look at sort of year to year, I'm still figuring out what to make of this. I'm curious what anybody else thinks about this, but maybe 2024 is kind of different. That's kind of my takeaway from this. And so I think this will be really interesting as we're able to extend this time series. We're able to look where the hope with PICO is to extend into over a decade, so we can have 10 years of data, and I think that will be a really exciting and rich data set to think about this provisioning. But overall, what we're kind of, what I'm gathering is that we're not seeing like a really strong impact. This intertidal eelgrass meadow declines didn't sort of crash the fish communities or drastically change what we see. And so this kind of raises our question, why are our fish communities remaining stable? We still have this habitat provisioning, even though the structure seems to have really changed. And the answer really is, I think, that the intertidal meadows are only one part of the seascape, right? So we're thinking about declines, and to us they're very obvious because we're sort of land-based, and so we're in the intertidal and we see these declines and they seem so dramatic, but these meadows are often adjacent to subtidal meadows that are very robust, and in these cases, these sites have not declined and have remained pretty robust throughout this time period. So this figure comes from some work we did with the Washington DNR where we used their data. So they have longer-term monitoring data sets they don't revisit as often, but they have over a longer period of time at more sites. And basically what this plot is showing is the places where, this is sort of the depth of their different sites. And so places where there have been declines tend to be shallower sites versus places where there's an increase tend to be these deeper sites. And most places the eelgrass isn't doing so well, even in the DNR surveys, but the places where it seems to be doing better seem to be these deeper sites. And so this idea that basically these subtidal meadows may be robust, they may be buffering what's happening in the intertidal, and so the fish communities are actually like, maybe they're fine. And this eDNA integrates over about a 1 kilometer radius. So maybe our sort of habitat provisioning is like pretty much okay, even though we see these kind of dramatic declines in the intertidal. So that's sort of interesting. What I think is even more interesting is whether this is going to continue to be the case as we sort of continue to experience disturbance from these marine heat waves or atmospheric events and more and more extreme temperatures. So this is a plot, again, of sea surface temperatures. The gold line is that 90th percentile, and the black is the observed temperatures. This is just for one site. But the event that I'm talking about that has had this really big dramatic effect is like early June 2021 here. You can see in the record, it doesn't even really show up in the record. And part of that is because it was an atmospheric event, not a marine event. So the sort of, it's a little bit apples to oranges, but we're continuing to experience these more and more extreme temperatures. And I think the question of whether these subtidal meadows, how robust are they, how much of a refuge can they continue to provide is kind of open. And I'm gonna just really quickly pause because there's a raised hand in the Zoom. I don't know if there's anyone else on Zoom. Maybe they don't, maybe, okay. I don't see anything in the chat, so I'm gonna let it go, okay. Okay, great, yes, so, yeah, so, we'll see. This is kind of a we'll see. I think we have to, this summer is gonna be interesting with the El Nino, our possible extreme temperatures coming. It'll be interesting to see. And now I've messed up. Clicker. Aha, okay, so to kind of bring this together for the seagrass side, this idea that these intertidal areas are very dynamic. They can experience these rapid collapses. They can sort of have this sort of extreme change over a very rapid period of time, but despite this kind of structural loss, these fish communities seem to be intact. We seem to maintain a lot of our function if we have this sort of buffering of the subtidal meadows. And I think that part of the takeaway for me is thinking about, especially when we think about applications, conservation, and restoration, we have to take a kind of landscape or seascape scale view and think about this integrated mosaic, and not just like the exact area where we put our transects that's covered in macroalgae this thick. Very sad. Okay, so that's seagrass. We're going to say goodbye to the beautiful seagrass meadows in the San Juan Islands, and we're going to say hello to also beautiful salt marshes in close Coos Bay, Oregon. So it's great to work with people who can fly drones, and they get you really beautiful imagery. Okay, so we're going to move kind of up in the tidal frame, up into the salt marshes, and thinking about switching as our function, thinking less about habitat provisioning, and now thinking about carbon seed restoration as this function. Key ecological function in salt marshes, and it's blue here. The C's are blue for blue carbon. So blue carbon. So people familiar with the term blue carbon? Yes, I know some people are very familiar. Okay, so this idea that marshes, along with other coastal ecosystems, can sequester a high amount of carbon relative to other ecosystems. And one of the really important questions to understand about blue carbon is, where does this carbon originate? So where does it come from? How fast is it accumulating? How much is it accumulating in these different ecosystems? And part of the reason for that is because of this interest in blue carbon as this kind of climate mitigation solution, or this sort of process that can help us with climate mitigation. And so there's been this sort of big interest and investment in thinking about salt marsh restoration as a way to help buffer climate change. There's this massive salt marsh restoration project happening in San Francisco, the South Bay Salt Ponds, biggest salt marsh restoration on the West Coast. Very exciting. But again, even so, to understand the sort of impact of this restoration, we have to know how much carbon is being stored, how fast is it being stored, where is it coming from. And the question of where it's coming from is really important because of this concept of additionality. So if we want to know that we restore the salt marsh and now more carbon is being stored there, we have to know, well, like, more compared to what? What's the baseline? Where would it be coming from? What would be happening to it otherwise? So we have to think about this idea of additionality. So this sort of like thinking about the benefit of a restoration in terms of how much carbon is stored compared to some baseline. And then we have to think about the source of that carbon, if it's local or imported, affects how we can kind of consider it. So if we think that that carbon is actually all forest carbon, it's fixed in the forest, it flows out to the marsh. If the marsh wasn't there, it would just flow out to sea and be buried there. We kind of can't count that as extra carbon being stored. So we have to sort of know something about where that carbon is coming from. And this is especially important, again, from this kind of applied standard if we're thinking about carbon credits and carbon accounting and sort of that whole side of blue carbon world. Okay, so where does marsh carbon originate? There's also very interesting ecological questions to understand about this. Like, it is interesting to know, is it all forest carbon that's being stored, or is it the marsh carbon? And there's sort of ecological implications of that. There's this carbon accounting motivation. And typically the way this has been done, the way this has been looked at, is using stable isotopes as markers to try and differentiate these different sources. And when we started looking into this question a little more, I was kind of surprised because in mangroves, so our other blue carbon systems, there's been lots of work done, there's been some nice synthesis, and we think about half of the carbon in mangroves is local versus imported, and the same in seagrasses. There's been some nice synthesis work, about half, sort of back of the envelope, generalizable. It's going to be site-specific, but in general, we'd say about half is local versus imported, but nobody has done this for salt marshes. So I kind of was like, well, someone should do it. There should be a number. We should estimate this. So we set out to do that as one part of this work. It's a big global literature synthesis, and you don't really need to read all this, but we looked at lots of abstracts. We extracted lots of details. I will say, metadata was so thin, like people explaining what kind of marsh site it was, very minimal. So always report your metadata. Always say like what kind of site you're working in, what's the salinity, what's the, I don't know, just, that would be very helpful. But anyway, and we're looking again, we're looking at 13C and 15N as our isotopes, and then C to N ratio as another tracer to understand where this carbon is coming from. And I'll just walk you through a little bit of this. So we have a global data set, and it is, as you can see, pretty spatially biased. We have a lot of North American values, we have some European values, we have some values from China. A lot of the world we have no values, which is sad. It's sad, but we did try. So The other thing is that we really have very few measurements with multiple tracers. So our whole data set had 13C. That was the minimum for us to include it. But when we try and get down to 2 tracers or 3 tracers, we really started to be just a few dozen measurements, which is sort of not that many. I was actually surprised. There's just not that many measurements being made. So that's interesting. We're going to look at a couple of stable isotope biplots. So just to walk you through this, if you haven't been thinking about stable isotopes recently, we can think about, in this context, we can think about delta 13C, so our stable isotope of carbon signal as moving from a more marine to a more terrestrial signal. For nitrogen, 15N nitrogen, we can think about this as often we think about degree of processing, if we think about trophic levels for that kind of work. But in this work, we can also think about a kind of marine to terrestrial gradient as well in 15N. And so I'm just going to start throwing data up here, and I'm going to walk you through it. But our data set, when we plot it, looks like this. So the points, the points are our measurements. So each of these points is a study where they went to a marsh, they took some sediment, they did the stable isotopes, and it's plotted up there on these 2 axes of 13C and 15N. And then the colors are from our sources. So this is like if you take some marsh vegetation, dry it out, grind it up, look at the isotopes, and then plot those values up there. Those are kind of our different sources that we might be drawing from. And so we have a number of different sources. Salt marshes are integrating, they're at that marine terrestrial interface, so they're pulling marine vegetation, salt marsh vegetation, and then freshwater as well. And so you can see our terrestrial signal is really clearly separated, so things like forest plants, upland plants, they pull out really well. Our marine signal differentiates pretty well, so seagrass and macroalgae, and then also salt marsh C4 plants, which we don't need to worry about too much in this context. They pull out pretty well, but then we have a lot of overlap in the source signal between marsh plants and diatoms and freshwater, sort of freshwater marine diatoms and things like that. And that is part of the challenge of using stable isotopes, is that they can only differentiate so much. So we ran some mixing models with this data set to see what we get, and what we get is a sort of answer that's pretty congruent with seagrass meadows and mangroves as well. We found about 40% of our marsh carbon comes from terrestrial sources. And this plot, these are probability densities, so this is kind of like the most likely distribution of each of those sources contributing. And so we can see that we have a pretty clean terrestrial contribution. And then our sort of other takeaways from this is that we have relatively low contributions from our marine sources. So we didn't have salt marshes aren't bearing a ton of seagrass or macroalgae, which also isn't that surprising. But we kind of don't have great resolution of our other sources, and we still have this question, because there's this overlap between our sources, and because this overlap includes things like diatoms, which can be local, they can be in the marsh, or they could be coming from freshwater sources or being brought in the estuary. We really can't answer this local versus imported question just with stable isotopes alone. So we have some limits to this global synthesis. And yeah, so these contributions from our terrestrial contribution we can kind of estimate, but our sort of differentiating other sources with isotopes alone is very difficult, and there's limited data available globally. So what did we do? How did we solve this? If you guessed eDNA, you would be correct. So our goal here is to use eDNA now in the soil. So looking not in the water, but looking in the soils. And the reason we can do this is because our soil cores are basically, they're time capsules, right? The deeper we go, the further back in time we're going. And so we can look at the accumulation of marsh sediments over time and look at the eDNA, the genetic material that's held in the soils, that's sort of stabilized there, and we can interpret that to understand what contributed that organic matter over time. And so what we've done is to basically subsample sediment cores where we look at both isotopes and eDNA from 2 halves of the same subsample, so that we're able to get really closely tied isotope and eDNA data, which is very exciting, and it's also taken a long time for us to get this to work. Some of what I'm going to talk about is part of Katrina Poppy's dissertation work. She's up at UBC in Western Washington, but she has been a main collaborator on this project who's done a lot of work in marshes across the Pacific Northwest. And again, we did this, have 600 sediment core sample. We're doing all this root removal. We did paired subsampling, lots of fun stuff. Okay, so this is Katrina's data set. This is the isotope version of Katrina's data set. So you can see, again, our sources. So we're sort of spreading out the sources across this continuum. And again, our terrestrials down here, marines over here. And of course, all the salt marsh signal is like in this middle part that's really mixed up with other sources. So we have the same challenge as with the global data set, is to distinguish these salt marsh sources, diatoms, other things. They're all overlapping. We're still, the eDNA is coming back to us in pieces. So I'm only going to show you a little snippet. We're going to zoom in on one marsh here, which is, this is a restored marsh. It's been restored for about 30 years. And I'm going to show you eDNA from 3 different depths. So at the surface, 10 to 15 centimeters below the surface, and 25 to 30 centimeters below the surface. And we know what the surface communities are. We know what the marsh communities are at the present time. And then as we go back in time, we can kind of look at those changes over time. And in these slides, so for our eDNA, these panels, we did a lot of methods testing. So these are kind of different extraction methods and different methods, development things. But what we ended up settling on was this sort of, what we have symbolized here as the E, but they're pretty consistent across all of our different methods. And so that's kind of our high throughput version. So we don't have to worry about that too much. But we can kind of look at these. We can go through the levels of the taxa from phylum, what's being dominated by vascular plants, which makes us happy. It's the marsh, so we like to see that. We can sort of keep going down a little bit further, getting down to our family, where we're mostly seeing most of the DNA that's present is coming from rushes, which are the marsh vegetation that's present. And so we think that this is a site that's dominated by local carbon. So that we can sort of identify which family this is. We're not seeing kind of conifers or other sources. And so this gives us a little bit of confidence to say that this is more local carbon as opposed to external. Okay. The last piece of this work is zooming in from this regional scale work down into one single marsh, where we can look at some of the variation across environmental gradients. So we can look at an elevation gradient across the marsh. And this is, again, this is work that my master's student Christine is leading. And this is the same isotope bioplot as before. We have our same sources up here. Again, terrestrials down here, marines over here, all of our salt marsh stuff is here. But what's sort of interesting, these are across a single marsh, an elevation gradient, and we see the same spread in isotope values as we saw across the whole Pacific Northwest, which is like almost as much as we see across the whole globe. So there's just a ton of variation in these isotope signals, which again makes it a little bit tricky to use them to really start to sort of hone in on these questions. One thing that's interesting, though, this is that same marsh, that restored marsh, and across our elevation gradient. So the colors here are, the red is this sort of lowest elevation, and then green is in the middle. The blue is the highest elevation, and then the purple is our reference site, which is an undisturbed marsh site. And so we do see across this elevation gradient the restored marsh sort of coming to resemble the reference marsh more. So that's sort of encouraging that we see that pattern happening. Because again, we're really interested in this carbon sequestration function, and if we can return that function with restoration, like when we restore marshes, do we sort of regain their ability to store carbon? And so one way that we need to understand that is, well, are they storing carbon in the same way that an undisturbed marsh or reference site would? And so this isotope data starts to give us some more information about that. And we do see this reestablishment of carbon sequestration function after restoration. And there's more eDNA forthcoming on this, hopefully. Okay, so just to give you an image, when I say the low to high, so this is, I showed this in the drone imagery, but this is like the high to low elevation in the salt marsh. It's really not that far of a difference, especially, but again, we have this like variation, incredible variation in the isotopes. And so we sort of need the eDNA to try and help us refine that a little bit. Okay, so my takeaway from this is that marsh carbon contains multitudes, and these multiple analytical methods really help us to kind of better hone in on the sort of specific dynamics of, we have this idea, marshes store carbon, but what does that really mean in different places? Where are they getting that carbon from? We really need this sort of different ways of approaching that to get at it. We see this, again, this variation within a site is almost as much as across the Pacific Northwest, which is pretty much almost as much as across the whole globe. So it's That's kind of wild to me, honestly. Maybe it means the global distribution is actually just really small and it's just one Marshall's worth. I don't know. We have to dig into this more. But I think that the hope with this work, I mean, there's lots of sort of interesting ecology behind it, but eventually our hope is to be able to better inform this carbon marsh restoration work, to better be able to understand how choices about land management, how choices about regrading marshes, changing their elevation for restoration purposes, does or does not have a carbon implication and a climate implication. Okay, so you might wonder if I think that eDNA is the answer to everything. And I do think eDNA is really cool. And as a field person, I think eDNA is great. And I do think, so I am like a big fan of the sort of multimodal investigation, taking different approaches. There are a lot of caveats to eDNA, which I have not explored here. I think probably people in this room are pretty familiar with some of those caveats and some of the kind of constraints. But I do think, so for me, certainly, this kind of work is only possible through collaboration, and I do think that this kind of science as a team sport is like, my sort of takeaway from doing this work is that we have to have this collaboration to make these kinds of methods work, and that is my high horse that I like to put in a little slide about, but like collaboration is it's a practice. You have to engage in it, you have to give resources to it, you have to do it to make it work. Yeah, and I think project infrastructure, which means a lot, is really key to making these kinds of collaborations work. And that includes like within a research group, within a university, but also these kind of big coast-wide, 15 sites up and down the coast, across all kinds of institutions. They're fun. I love them. They do take work. So I think I maybe kind of talked fast. I don't know. Lots of people to thank. Thank you for listening. I'm happy to take any questions.
Thank you, Lillian. That was awesome. I'm going to also grab a second mic, I think, because we want to make sure folks on Zoom can hear, and maybe Serena can be the second mic person. But we'll take any questions, and I will come to you with a microphone, and you'll have to speak into the microphone if you ask a question. But don't let that deter you. Okay, Claire?
That was really cool. For the fish eDNA and the seagrass, so there looked like there was variation across those sites in terms of the habitat, even post the heat wave. Do you see any relationship between like intertidal habitat structure and the fish that you saw, or is it just like, I haven't found one.
No, we haven't yet. Like we've looked and we haven't. What I haven't done, in terms of like sort of raw metrics like density or cover, like we haven't found anything. What I want to look at is, which I haven't done, is sort of recovery metrics. So like over time, if we see like time to return is smaller or sort of like percent recovery is smaller. I think that, but again, I think because the eDNA integrates over such a large area, I actually might be surprised if there was a really strong signal, so.
Hey, Leland. So first question is, what fraction of the beds that you see, the eelgrass beds that you see in San Juan, would you say are intertidal? So the intertidal is all gone. Like, is that 10% of the eelgrass? Is that 50% of the eelgrass? 1% of the eelgrass?
I think it's probably closer to maybe 10 to 20. It kind of depends on the site. It's certainly not 50%. So
Okay.
But most of our subtidal beds are really poorly mapped. Like we don't really know where the bottom edge of it is, except for like boating over it as we come in, right? So, but yeah. So again, it's kind of like maybe it's kind of a no duh. Like the intertidal climb is just this little strip, and the subtidal's totally fine, and it's way bigger, and so everything's totally fine there, Yeah, I think it's probably closer to 20%.
The follow-up then is just sort of like, well, what do you think? I mean, I know for something like eDNA, the scale is too big to distinguish intertidal versus subtidal like functioning. But other things that maybe you or others have mentioned, have measured, like can sort of be measured on a more local scale. I mean, is there a difference in the functional sort of role or value of intertidal versus subtidal?
So that is such a good
beds?
question. Yes, and I think there is a lot of variation in plant traits. So like sort of obvious things like morphology, like the size, the subtidal plants are way bigger. But we think that the growth is also different, like how fast they're growing and how sort of fast they turn over, which does relate to disease dynamics. So yeah, there is part of that. But also, yeah, their reproductive strategies seem to be like the sort of density and timing of seed production is very different. And so if we're thinking about things like collecting seeds for restoration, for example, or even just like the capacity of these beds to reseed themselves, like the subtidal is maybe has less reproductive capacity. And so thinking about those kinds of trait differences, or I don't know, I'm sort of hesitant to say that. But yeah, I think there are a lot of differences. And I think if we think about kind of the other sort of, this sort of like, ecosystem metabolism questions, is like the intertidal, because it's growing so much, may actually be sort of up to more, like doing more in that sort of ecosystem metabolism way than the subtidal is. So yeah, it's a really good question. I think, so these sandworm beds, I don't think they're storing very much carbon because it's very sandy and they get a lot of hydrodynamic energy. So I don't think they're like accreting very much. But I do think, I think if you lose those intertidal areas, you are going to lose carbon, I think. So.
Nope. Okay. I was really interested in your isotopic carbon graph and your last point about the elevation of restored meadows eventually reaching the natural meadows. And I was curious if that graph was showing us both magnitude of carbon stored, type of carbon stored, or both.
Yeah, it's mostly type. So it's not magnitude. We do have that data for these areas, and I don't know off the top of my head. But the high marsh is probably storing more carbon, but less densely than the low.
And so if it's the type of carbon, was the lower elevation marshes storing more things from the sea?
Well, maybe. So they might be. It might also be that they're, I don't know, there's a lot we still have to unpack, but it could be more of a kind of diagenesis, like there could be just more microbial stuff happening in those lower, sort of there's less vegetation. And it could also be that the vegetation is really just trapping more, so we're just getting a higher terrestrial signal. They're all storing the same amount from the ocean, but we just have a higher terrestrial signal because it's trapping more, and so then it sort of like shifts it, but yeah. Lots to think about. We're still thinking about it, but yeah.
Is it on now? Oh, yeah. Super cool talk. For the PICO stuff, have you looked at the fish community like through a functional analysis? Like, what are the functional traits represented? Are the communities different from, the communities look very different, or the southern and northern, or whatever the differentiation between there's one group and another? Do you know, like, functionally they're made up of similar fish or different fish? Or
yeah, so no, the short answer is no. I think it would be good to do that. I need someone who knows about fish or who wants to learn about fish to do that. But I do think that the PICO stuff is interesting because we can put our site in the context of all these other sites up and down the coast, including here. And I think we do see reduced biodiversity in the San Juans compared to other places. And so whether that, like, how much the variation between like our spatially distributed sites relative to kind of eelgrass in the Pacific Northwest or eelgrass on the West Coast is probably like a pretty small amount of variation, even though they kind of break out spatially. But yeah, I think PICO, there's a lot more coming from PICO very soon. So.
I have a question. I have a question about timescales for the coring. How does the timescale of like eDNA kind of removal from a system or like residence time in the system, how does that compare with the way that we think about carbon sequestration or like carbon values in the system? Like, like if they're in the same layer, can you think of it as like the same time scale, or is there some offset?
What a great question. Yeah, so
I think it's super cool, but I'm
no,
like,
sediment
oh, yeah.
eDNA world is like full of these questions because we sort of don't really know the answer. Like we can, so DNA can persist for a really long time. So we have like ancient DNA, which is sort of different from what we're doing, but like you can sort of extract very old DNA from sediments as well as from like artifacts and sort of archaeology, right? Which is very cool. Yeah, this question about like, do we see like, if we go 15 centimeters down and we think that's 100 years ago and we see eDNA there, like, is that from 100 years ago or is that from something else? Like, that's a really important question. And one way that we're able to kind of have some confidence in what we're doing is because we did the stable isotopes at the same depths. And so if we see like agreement between those methods, we're kind of like, well, we believe the stable isotopes. So like, hopefully if they're matching the DNA, then we can kind of have some confidence. But yeah, it's a tricky question. The other thing, I guess, one thing that people are concerned about sometimes is like inputs. You know, the plants are growing their roots down, and so they may be just putting like fresh DNA in there all the time. We took the roots out, which was a lot of work. So at least the macro parts are removed. But whether they're sort of like fresh exudates, like that's a tricky question.
Oh, I think it's cool though if like you can do that, and I'm wondering if it's with like the seagrass system, if there has been periods in the past where there's been a switch from eelgrass to ulva or other macrophytes. Like, would you be able to detect that in a core?
Yes. So I don't know if in our systems where we've had these very short-term it depends on how long those persist, right, and how long the sediments are accumulating. But there is work that has been done in other places where they've looked at deep cores, and you can see the place I'm thinking of is the Virginia Coast Reserve, where I did other work before, but they've done deep cores and looked at like seagrasses that used to be marsh, like there was island turnover and they were marsh, and you can see like a meter down, it's marsh organic matter, and then it's seagrass further up, and then you can see the gap when it was lost, and then restoration. So you can reconstruct these histories, yeah, which is really cool. So yeah.
Okay, any other questions? Great. Well, thank you so much, Lillian. Another round of applause. We appreciate it. Come back again.